Obeticholic acid in NASH. Interim analysis from Regenerate trial. Non-alcoholic steatohepatitis, that is NASH, is a progressive form of non-alcoholic fatty liver disease that can lead to cirrhosis. In NASH, there is lipid accumulation with evidence of cellular damage, inflammation, and different degrees of scarring or fibrosis of liver tissue. NASH is expected to become the leading cause for liver transplantation, and most importantly, still there is no approved drug for its treatment. This creates a major need gap to be filled. CDCA, that is chenodeoxycholic acid, is primary human bile acid, and is an endogenous ligand of farnesoid X receptor, that is FXR. OCA, that is obeticholic acid, is synthetically modified analog of CDCA. Obeticholic acid is 100-fold potent than CDCA. Regenerate is a multicentre, randomised, placebo-controlled, phase 3 trial. Regenerate trial evaluated obeticholic acid in patients with NASH. Total of 1968 patients were randomized by 1:1:1 ratio to receive obeticholic acid 10 mg, obeticholic acid 25 mg, or placebo. Patients received treatment orally, once daily. The key inclusion criteria for patient selection were: age of 18 years or more, has definite or borderline NASH, with NAS more than or equal to 4, fibrosis stages F2, F3, or high-risk F1 stage, and at least one comorbidity. Patients were excluded if there was presence of cirrhosis, other chronic liver disease, elevated alcohol consumption, or there were confounding conditions. After randomization, interim analysis was carried out at 18th month. The study is ongoing with event-driven end of study. In this analysis, total 931 patients, with stage F2 and F3 fibrosis were included. This intent to treat population was distributed as 312 patients from OCA 10 mg group, 308 patients from OCA 25 mg group, and 311 patients from placebo group. Primary endpoints of this interim analysis were set as fibrosis improvement by at least one stage, with no worsening of NASH, or NASH resolution with no worsening of fibrosis. Study was considered as successful if either of the endpoint was achieved. At 18th months, study outcomes showed that fibrosis improvement endpoint was achieved by OCA at 25 mg as well as at 10 mg dose. 12% patients in placebo group showed the improvement in fibrosis endpoint, while 18% patients in OCA 10 mg showed the improvement. The significance was more in OCA 25 mg group, with improvement in 23% patients. NASH resolution endpoint was not met at time of interim analysis, but more patients in OCA 25 mg group showed at least 1 point improvement in scores in key histological features of NASH. These involves lobular inflammation, which was improved significantly in OCA 25 mg group compared to that of placebo, and the hepatocellular ballooning, which was also improved significantly in OCA 25 mg group. No new safety signals were reported during the study period, other than previously known adverse outcomes. In this study, pruritus was most common adverse event, occurred dose-dependently, with maximum events in OCA 25 mg group. Incidence of serious adverse event was similar in all the treatment groups, with no significant differences. Regenerate study is a step towards establishing obeticholic acid for treatment of NASH. It is the first phase 3 study in patients with NASH, with positive outcomes. Obeticholic acid 25 mg significantly improved fibrosis by at least one stage with no worsening of NASH, and it also improved key components of NASH disease activity, that is, lobular inflammation and hepatocellular ballooning. Overall, treatment of NASH patients with obeticholic acid was associated with clinically significant histological improvement that is reasonably likely to predict clinical benefit. Study is ongoing. Long-term outcomes may provide strong foundation for obeticholic acid to be the first approved treatment of NASH. The full text of the published article can be read at journal website. This video is for educational purpose only. We don't have any conflicts of interest.